Hey, I'm Klaus Wunderling, and as a postdoc in the group of Professor Thiele in the Leibniz Institute from the University of Bonn, I worked on metabolic tracing from alkyne-labeled lipids, focusing on the triglyceride metabolism from hepatocytes and adipocytes. I'm also the first author of the publication Triglyceride Cycling Enables Modification of Stored Fatty Acids. In the next 2 minutes I'll tell you a bit about it. Our publication features a mass spec-based tracing technology for alkyne fatty acids, providing experimental data which shows for the very first time that the intercellular triglyceride pool from 3T3 cells and adipocytes is subject to a constant cycle of degradation and reacylation, resulting in a rearrangement of all the fatty acids with an estimated half-life of 2 to 4 hours. Our findings emphasize the function of the TG cycling for maintaining the composition of the stored fatty acid pool in adipocytes. The processes we describe here contribute to a more differentiated view on how the adipose tissue could act as a supporting organ, functionally maintaining the fatty acid composition of the whole organism. For me, the most important aspect is for sure the adaptation of our tracing technology to now being able to simultaneously trace 3 different alkyne fatty acids at once. With that, we could now properly address the question on whether and how exactly the intercellular pool of triglycerides is cycling in adipocytes. With that at hand, I find it quite astonishing to follow the metabolism of 3 different fatty acids over a time course of up to 48 hours. Personally, figure 7 is for sure my favorite one, since my colleague Jelena Zurkovic and myself need to put a lot of effort in generating the dataset from this figure. But in the end, we could nicely show with this dataset that the cycling process we describe is not only present in 3D cells, but as well happening in primary white and brown adipocytes. Both cell types show cycling rate comparable to those we describe in 3D cells, and in the end it rounds up the paper quite nicely by adding a dataset from a primary cell culture, further highlighting the biological relevance of the findings. We hope that our publication provides further insight for the scientific community to better understand the intercellular triglyceride metabolism in adipocytes, both in adaptation to changes in energy consumption as well as its virulent thermogenesis. This might ultimately lead to treatment for patients suffering from the pathophysiological aspects of obesity. Furthermore, since our tracing technology is not only limited to a certain cell type, a distinct fatty acid or lipid molecule combination, or the aspect of triglyceride metabolism itself, we hope that we can provide future collaborators a platform with which they can address their questions on project and lipid metabolism involving the need to simultaneously trace lipid molecules.